Sydney, where are you originally from?
I grew up in Massachusetts.
Nice. Whereabouts?
Kind of on the Mass Rhode Island border. So I'm
Oh,
like
I know
20
where I'm from, New
across.
Hampshire. So
Yeah.
I used to live in Rhode Island, so I'm very familiar with it. Just a few students from you guys from Massachusetts and Rhode Island, New Hampshire and Maine.
hmm
So, but people are just going to start coming in here any minute. We got you'll start seeing the attendees list in there. So I'll put a public service announcement in just a minute or two. Good evening, everybody. Just letting you know that there's a few more people popping in. We're going to start right around 6:01. But tonight's lecture, we'll open up for questions as you start to have questions throughout the presentation, or just want to put some feedback or comments. Please put them in the Q&A or chat box, typically the Q&A section. And at the end of the presentation, we'll be happy to answer any questions you have as well. A few more students coming in at Sydney, so I just want to give it another minute. Looks like, Sydney, we're going to open it up here. Thank you, everybody, for coming tonight. My name is Matthew Adams. I'm the graduate director here at the Halmos College of Arts and Sciences. Work with students in incoming for any of our graduate certificate, master's, which is in Master of Science, our MPS, our MA, as well as our PhD programs here at the college. So we run this virtual lecture series with our faculty and graduate students. Sydney's the first one for the fall of 21 for our graduate students to participate. So we're super excited to have you here. She's a second-year graduate student here in the MS in Marine Science graduate program in Dr. Kerstetter's lab. His lab is the Fisheries and Avian Ecology lab here in the Shore Building on the Oceanographic campus in Dania Beach, Fort Lauderdale. So just letting you know too, our research focus is on the age and growth of mesopelagic fish. And also just want to let you know that Sydney was also one of our undergraduate students. She graduated back in May of 2020 and then started her graduate program that fall. She doesn't, she basically just, this is like one of the beginning of her graduate career, but she's in her second year and she's been doing some extensive research and working in Dr. Kerstetter's lab. So she's going to open up for questions at the end of the presentation. I will also be available to answer any questions regarding any possible questions on graduate programs. But for the most part, this is just a virtual lecture tonight on mesopelagic mysteries, understanding the deep sea fish populations through age and growth analysis. So I'm going to turn this over to Sydney. Thanks again for starting this off, and we're looking forward to hearing your presentation tonight.
Yeah, absolutely. I'm excited to share with you guys. Alrighty. Awesome. Just move that. Perfect. All right, guys. So tonight, as Matt said, I'm going to be talking about the mesopelagic environment, all of the characteristics that make it such a unique environment and makes it difficult to study as well. And then I'm going to be highlighting some other labs on our oceanographic campus that do research with mesopelagics. And then I'm going to talk a little bit about my current research project that I'm doing for my master's thesis. So just to kind of introduce myself a little bit more, as Matt said, I'm in my second year in the Master of Science in Marine Science program, and I'm pursuing a concentration in environmental science. I'm in Dr. Kerstetter's Fisheries and Avian Ecology lab, and I was lucky enough to begin that professional relationship as an undergraduate. And as an undergrad, I also had the opportunities to do a couple of really cool internships. So I spent a summer back in my hometown volunteering at a U.S. Fish and Wildlife fish hatchery. And I spent a summer up in New Hampshire working on a whale watch vessel. And then the past couple summers, I've been working in marine education. But it was really that experience at the fish hatchery that made me want to pursue a career in fish biology and fisheries science in general. So once again, I graduated in May of 2020 from undergrad at NSU with my Bachelor of Science in marine biology and biology with a minor in marine ecology. I was also part of the dual admission program, so that made my transition from undergraduate to my graduate career a little smoother than I know a lot of other people experience. And besides being a full-time student and working on my master's thesis, I'm also a lab assistant. So I set up and help with some of the intro to marine bio labs, as well as Dr. Kerstetter's biofishes lab. So to start off with a little bit of background about the mesopelagic environment, overall, the conditions are deep, dark, and cold. And because of these pretty extreme conditions, we see a lot of adaptations in the organisms that live in this environment that help them to not only survive in these conditions, but also thrive in them. So the mesopelagic environment is the zone of the ocean that's typically between 200 meters and 1,000 meters in depth. And then we have the epipelagic above that, and then the bathypelagic below. So all of my friends that scuba dive know that as you get deeper in the water column, pressure increases. So typically with every 10 meters in depth, you increase by 1 atmosphere of pressure. So right now we're all sitting basically at 1 atmosphere. So even diving down just 10 meters or about 30 feet, you experience pretty much twice the amount of pressure that you would experience just sitting at home on your couch. And so when you get down to that 200-meter mark or even that 1,000-meter mark, some of these organisms are experiencing 20 to 100 times the amount of pressure that we experience on the daily. And so for any organisms that aren't meant to be there, that can be literally quite crushing for their body structures. But all of these organisms have adapted to be able to protect their proteins from being crushed. So proteins are a really important part in our body's functioning. So they have to be able to protect those proteins. They need to be able to stay neutrally buoyant in the water column. So that means they're not going to naturally float or they're not going to naturally sink. They want to kind of stay right where they are without having to exert energy to do so. And then once again, kind of maintaining that body structure. So I'm sure a lot of people have seen pictures of deep sea organisms like a blobfish or even orange roughy that are brought up to the surface. And you can see that their tissues are totally distorted. Some of them have their insides exuding from their mouths. And all of that is because of the really quick pressure change that their body doesn't have the opportunity to acclimate to. And the next condition of the mesopelagic environment is that it's really pretty dark. So the mesopelagic environment is also called the twilight zone or the aphotic zone. And this means that less than 1% of natural sunlight reaches these depths. And even when there is light at these depths, it's going to be in the blue and the purple wavelength spectrum. Red light and orange light, those longer wavelengths, are typically the first to be absorbed. And that kind of ties into some of the adaptations that these organisms have. So the first is bioluminescence. And so bioluminescence can occur in 2 ways in the deep sea. The first is symbiotic bacteria. So I'm sure everyone's seen what an anglerfish looks like. They have the lure on their forehead, and that is basically a modified dorsal fin that holds a stack of bacteria that can glow. So anglerfish are known for their symbiotic bioluminescent bacteria, while lanternfish, like in the photo on the right, they have cells that are called photophores, and those cells can actually be controlled by the fish itself. So they can choose the pattern with which they blink or how long they glow. So those are the 2 ways in which bioluminescence occurs, and typically about 90% of the animals that are in the mesopelagic and the bathypelagic are able to bioluminesce, which is really cool. The second adaptation for these dark conditions is body coloration. So going back to kind of that red and orange light that gets absorbed, some organisms have adapted to have a red or an orange body coloration. And while this can be energetically costly to create those pigments, it allows these animals to become practically invisible in the deep sea. However, because it's pretty energetically costly, most of them are going to stick to kind of black, brown, gray, even silver body coloration. And then the final adaptation is really big eyes. So in the mesopelagic, where there's still, oh, the light went out. Sorry, give me a minute. I think it's motion censored. Okay. Where was I? Okay, big eyes. And so in the mesopelagic, because there's still a minuscule amount of light, those eyes are going to be as big as they possibly can to try to absorb as much light as possible. So for example, that lanternfish that I talked about earlier, even though the fish is pretty small, you can kind of see just like the proportion of the eye size to the entire head size is pretty massive. And even with the barrel eye fish, where the front part where you would think that's where their eyes are, these are actually olfactory organs, so they help with smell. And their eyes are actually these really big green organs here that can face upward to be able to see shadows passing over. And this is a pretty unique trait to the mesopelagic environment, because once you hit that bathypelagic environment and you have absolutely no light whatsoever, it's not worth it for these organisms to put energy into creating large eyes. So in the mesopelagic, there's organisms with big eyes, but in the bathypelagic, we're going to see organisms with much smaller eyes or really reduced eyes in general. And the last thing is that the mesopelagic environment can get pretty cold. So typically above 200 meters in that epipelagic zone, the temperature is going to stay relatively constant. And I say relatively because there are going to be seasonal changes that can cause that thermocline to become shallower or become deeper. But relatively, that epipelagic zone is not going to have a whole lot of temperature fluctuation. And then even below 1,000 meters, the temperature is going to kind of level off as well and stay relatively constant. But it's in between those, right in the mesopelagic zone, that we see this thermocline, where we see it go from kind of warm to a whole lot colder. And even though the exact temperatures in this diagram aren't super important, throughout the year, it could be like a 15 to even 20 degree difference between that 200 meter mark and that 1,000 meter mark. And so that's really important for fish that, or even other organisms that travel from the mesopelagic zone to the epipelagic zone, because now they are experiencing a wider range of temperature, oh my goodness, a wider range of temperature than most other organisms experience in their entire lifetime. So after all of that, besides giving us really crazy, cool looking organisms, why do we care what happens 200 meters below the surface? Like, why is this important to us at all? So mesopelagic organisms participate in one of the largest migrations by mass, and this is a daily vertical migration. So during the daytime, when there's a lot of visual predators up in the epipelagic, the mesopelagic organisms stay down in the deep sea. And then when the sun goes down and those visual predators kind of go back to hide, the mesopelagic animals come to the surface to feed at night. And so this is really important in our overall carbon cycling in the ocean and carbon sequestration. So the biological carbon pump is essentially what this is. And so this is when carbon or energy at the surface of the ocean is then brought down to the mesopelagic. And then in the mesopelagic, those fish or those animals might produce waste, or they might die, and then that trickles down to the bathypelagic. And so that's how energy is transported from the surface, where we got a lot of primary production, all the way to the mesopelagic and the bathypelagic, where there's practically nothing but marine snow. And so this also has consequences, I guess, for when there are changes in prey population sizes at the surface. So if we overfish a really popular prey item for these mesopelagic animals, then they're going to experience a food shortage, which is going to in turn change the way our oceans cycle its carbon. Additionally, we can have quite a few direct effects on the mesopelagic environment. And so the organisms or the fish that I'm working with are a significant portion of longline bycatch. So I have a little diagram there showing basically what a long line looks like. So we've got the floats and the main line, and then the individual lines with the hooks. And so back in the mid 1970s, there's a push for deeper set long lines. So instead of long lines laying closer to the surface, they weighted those main lines. And so they laid at depth. And this helped to prevent like marine mammals like dolphins and porpoises from getting entangled. It helped to prevent sea turtles from grabbing onto the hooks. And it actually also helped to exclude seabirds from their accidental bycatch. So while it helped in a lot of other conservation aspects, these hooks and these lines are now in the way of these mesopelagics that are doing their daily vertical migration. And so when these fish come across a baited hook on their way up to the surface, they're going to grab onto it. And initially, a lot of these species were considered pretty useless. They were bycatch, they were accidents. There wasn't a whole lot that these fishermen could do with them. So they would just toss them overboard. But over time, the more and more of these species that they caught, they started to gain economic and commercial value. So one of the fish that I work with, Escalar, they are actually sold for profit and consumed by humans in the US. So as these fish start gaining more and more commercial value, there's going to be less motivation to protect them for conservation efforts and to try to exclude them from the bycatch. So it's really important that we understand their physiology, their life history, just overall doing some fish stock assessments so that we understand the populations that may quickly become overexploited by accident, not even by directed fisheries. And so before I go into my own research, I wanted to talk about some other labs out at the OC that are doing mesopelagic and bathypelagic work as well. This is by no means an exhaustive list. I'm just listing a few faculty members that I've gotten a chance to get to know by taking some of their classes. So the first I wanted to highlight is Dr. Tracy Sutton and his Oceanic Ecology Lab. So Dr. Sutton is the director and the PI or principal investigator of the Deep End Project, which stands for Deep Pelagic Nekton Dynamics of the Gulf of Mexico Consortium. Quite a mouthful, but this is only one of the many initiatives and consortiums that Dr. Sutton is a part of. And so his lab focuses on the ecological structure of deep sea ecosystems, global deep pelagic biogeography, food web structure and trophic interactions, benthic pelagic coupling in the deep sea, physical oceanographic drivers, as well as taxonomy and systematics of the deep sea. So Dr. Sutton also teaches a couple classes at the graduate level. I'm currently in his ichthyology course, and I believe he also teaches tropical fish biology. So anyone out there looking into the grad program, Dr. Sutton is a really involved faculty member. The next lab I wanted to highlight is Dr. Milligan's Seascape Ecology Lab. So she is also part of the Deep End Consortium and is also part of the Delos Project, which studies abundances of deep sea organisms over different spatial and temporal timeframes off the coast of West Africa. So overall, her lab focuses on identifying spatial and temporal distribution patterns of these deep sea organisms and why they follow those patterns. Understanding how the migration and dispersal of deep sea animals creates connections. So that kind of also goes back to and ties into that vertical migration. So how these animals can connect multiple food webs, as well as understanding how deep sea ecosystems are impacted by anthropogenic activities. So how we as humans, how our impacts are how our activities impact the deep sea and how we can predict and manage that in the future. So Dr. Milligan currently teaches marine ecosystems at the graduate level, so I'm in her class. And then I also LA for her Introduction to Marine Bio class for undergraduate. And finally, the lab that I'm a part of, Dr. Kerstetters' Fisheries and Avian Ecology Lab. So a lot of the funding for our sampling has come through the Perc Project, which stands for the Pelagic Ecosystem Research Consortium. So we have a couple grad students that go out on these longlining vessels, and they bring us back our samples. They collect biological information, like the length and the weight of those samples. And so with these samples, we're doing a couple different studies. So Ellen is doing a diet analysis of lancet fish using the stomach contents and stable isotopes. And then Alexander is studying endoparasites in the lancet fish, as well as the gampillid fish. And so he's looking at the gills, the GI tract, the heart, the liver, and just overall the body cavity for these endoparasites, which is pretty cool. And then that brings us to my project, which is the age and growth of the Escalar, oilfish, and snake mackerel. So as I mentioned, I started my professional relationship with Dr. Kerstetter as an undergraduate. And at that time, he approached me with a project that he thought I would be a good fit for. So back in 2008, 2009-ish, he had a graduate student that had originally started this age and growth project, who unfortunately couldn't complete the project. So this data had been sitting around for about 10 years. So in the fall of 2018, I took on this project, and I've been working on it ever since. So when I took it from Carrie, she had about 100 otoliths in total. And even today, I was extracting some otoliths. So up through current progress, I have approximately 350 otoliths between the three species. And so I've been able to collect a lot more data than a graduate student typically would in that 1 to 2 year thesis timeframe. So this is, I'm hoping to be a really, really impactful study with all that years of data. So the fish that I'm studying, Escalar, oilfish, and snake mackerel, they are all part of the Gampillidae family. So this family is known for being large, swift predators, and there's about 24 species in this family. There's not a whole lot of biological information known of these fish, besides what we learn from incidental interactions with fisheries, whether it be trawling, longlining, gill nets. We don't really know a whole lot about them until we bring them up and study them. And then the reason we chose these species to work on is because they're such a significant portion of Longline Bycatch. So the first fish that I wanted to introduce you guys to was the Escalar. So this is one of the more common species that gets pulled up. So I have the most samples of these guys. And they're found in tropical and temperate oceans worldwide, typically between 40 degrees north and 40 degrees south. And they're assumed to be highly migratory. So as I going back to that daily vertical migration, Dr. Kerstetter actually a while back put out a paper where they used a PSAT tag, or a pop-up satellite archival tag, attached to an Escalar to actually track that daily vertical migration. So that using this satellite tag, they found that during the day, the Escalar spend their time at depths deeper than 250 meters. And then at nighttime, they come up to waters shallower than 150 meters. So they got tagged evidence of this daily vertical migration, which is really cool. And during the nighttime, when they're up at the surface, they're feeding on squid, crustaceans, and a variety of fish in this low light environment. And this photo does not do it justice, but in life, or very soon after life, these fish have really gorgeous iridescent eyes that are kind of greenish. And so that helps them to pull in as much light when they're foraging or they're hunting in these low light environments. These guys can reach a maximum standard length between 150 and 200 centimeters. And for everyone that does not automatically think in metric, 200 centimeters is about a 6.5 foot fish. So they can get pretty massive. And they're believed to reach sexual maturity around 30 to 35 centimeters, but we don't know how old that is or how long it takes them to get to that 35 centimeter mark. So that's just kind of reinforcing how important it is to understand the growth rates of these animals. And as I mentioned earlier, despite there not being a direct fishery for Escalar, they're pretty common bycatch. They make up 1% of the catch of tuna longlining, and they are sold for profit and human consumption in the US. So the next species I work with is the oilfish. Once again, widely distributed in tropical and temperate oceans and pretty migratory, except these guys prefer more benthic habitat. So they're still in that mesopelagic zone, but they're going to hang out more towards the seamounts or the continental shelf or the continental slope. And their bodies are covered with these spiny, bony tubercles kind of between their scales, which makes their skin really rough to the touch, kind of like sandpaper. And they use this to their advantage when they are hunting. So a study using a submersible observed oilfish hunting, and instead of darting and catching the prey with their mouth, they almost kind of body slammed their prey, inflicting a severe blow. And that actually, not only does it stun the fish or the squid that they're eating, but it also really scrapes them up. And so a follow-up with that was a stomach content analysis. And they actually found squid that had scraped up skin whole in the oilfish stomach, which is really cool to kind of be able to put those two pieces together and understand their behavior with that. These guys can also get pretty big, reaching maximum standard length between 150 and 300 centimeters. And none of the fish I study have a swim bladder. So a swim bladder is kind of just like an organ that holds gas that they can control to maintain that neutral buoyancy. So instead, very fitting for its name, they take the oils that they cannot digest from their diet and they store them in the form of wax esters in their white muscle and then in the roofing bones of their skull. So they're pretty, fitting for its name, pretty oily fish. And so when humans ingest oilfish and escalar, there's typically some pretty bad gastrointestinal side effects to that. And last but not least are the snake mackerel. And there's significantly less biological information on these guys. And these are also the fish that I have the fewest samples for. Once again, found in tropical and temperate oceans worldwide. They were previously shown to reach about 60 centimeters with a few individuals getting longer than a meter, but a lot of my samples are actually more towards the 80 to 100 centimeter mark. So that may have potentially just been some sampling bias in the past. They have these large fang-like teeth, you can kind of see in the photo, and they hunt squid and they like to dart after flying fish. So a boat at the surface at night was able to observe these snake mackerels darting after flying fish and catching them in the air, which is pretty cool. And like most mesopelagic or deep sea organisms, they tend to be solitary just because it takes, it's gonna be a lot more energetically costly to travel with a companion that might end up being your direct competitor for a food source. So they tend to be solitary animals. So when I was introducing my research, I mentioned otoliths. And so otoliths are typically the most common way that we age fish, but you can also use vertebrae, fin rays, opercular bones, as well as scales. But otoliths are typically used for aging. And so otoliths are sets of structures that are found in otic vesicles at the base of the skull of many fish species. And the morphology of these otoliths are different between species, but they are pretty consistent within that species. So the photo on the left, this one over here, this is a pair of Escalar otoliths, and then the pair on the right are snake mackerel otoliths. So within a fish, there's going to be three pairs of these otoliths. There's the lapilli, the sagittae, and the asterici. And all of these function as part of the inner ear sensory organ. So they help the fish to hear and to maintain balance in the water column. Sagittal otoliths, or the sagittae, are typically the largest. They're typically the earliest formed with the widest and most visible increment bands, which is the important part for aging. So sagittal otoliths are typically the more common otoliths that are used in aging studies, including my own. But if the sagittal otoliths are too small or they just can't be found, the other 2 pairs can be used for aging as well. And this is a photo of an oilfish otolith. So these otoliths form through continuous accretion of aragonite, which is just a form of calcium carbonate, and otolin, which is a protein. And so we get this alternating band pattern of mineral deficient zones and mineral rich zones. So in this photo, that translucent zone is a mineral deficient zone. And so those are typically indicative of faster growth, which just means that as the otolith grows, the minerals are being deposited in a thinner layer over a larger space. And then the opaque zones are typically slower growth, once again, meaning that the minerals are just being laid down kind of right on top of each other because the otolith isn't growing as fast. So they're going to be a more dense layer. And that creates this really nice banding pattern. And this banding pattern can occur even on a daily basis. So for a lot of these organisms that are going through that daily vertical migration, we might see these daily bands occur. But typically what we're looking for are annual bands that are called annuli. So in a lot of fish, we assume that the translucent and the opaque zone together indicate kind of one cycle of growth or 1 year. So to the fun part of how I get these otoliths out of the fish. So the official term for the method I use is the guillotine method, which basically means I make a transverse cut or a cut down through the head, all the way down through the operculum and the gills. And you can kind of see that on the photo in the photo on the left. And by doing so, I expose kind of the back end or the posterior end of the capsules that have the otoliths within them. So I put a blue circle around the left otolith, and then you can see the matching capsule on the right of the spinal cord as well. And so after finding the otoliths, which is one of the hardest parts, the next hardest part is getting them out without breaking them. So a lot of times I have to be really gentle, kind of working them back and forth. And then when I do get them out, you can see in the middle photo, they have sort of a membrane around them. And so after extracting them, I remove the membrane and I clean them off with DI water. And then I let them air dry, just so that when they're stored in the vials, they're not going to stick to each other or potentially break. So that just kind of ensures that they're ready to be stored and there's no special process for drying them out necessarily. And so after extracting them and after they're stored for a while, I embed them in little blocks of epoxy resin. And so by having them inside that block of epoxy, it kind of stabilizes them so that when the saw goes through the epoxy and the otolith isn't going to wiggle around. It's going to cut through exactly where I want it. And it kind of gives it a medium as well to be mounted onto a slide. So these otoliths these were just ones that I used for practice. These were just snapper otoliths. But you can see just how thin those sections have to be for the light from the microscope to be able to shine through and accurately show those light and dark bands. So typically when I'm sectioning, I try to aim for sections that are 0.3 to 0.5 millimeters thick. So my spacer between my two blades on the saw is 2 pieces of tin foil put together. So these are really, really paper thin sections that get mounted onto slides, and then we use a microscope with a camera to be able to observe these banding patterns. And you can see, so this darker area in the middle, this is the nucleus of the otolith. And so this is kind of where all growth originated from. And so if you section at a certain part of the otolith that doesn't have the nucleus, you're not going to get a super accurate reading of the age because you might be missing some of those primary bands. So that first, maybe even second year band, you might be missing if you don't section right through the nucleus. And unfortunately, I have not gotten to the stage where I have started aging my own samples, but there are a few challenges to aging mesopelagic otoliths. So a lot of these assumptions are based off of tropical fish and kind of tacked onto mesopelagic fish. So overall, their environment is not going to experience seasonality. So even though we don't know exactly why these bands are laid down in the patterns that they are, we can typically assume it's some sort of energy trade-off. So at some point in the year, there might be a food shortage, or there might be spawning, or there might be a large migration where they have to, all that energy that's typically used for growth of the fish has to be diverted to something else. And so that's kind of how we gauge whether or not these fish are going to lay down these increments in their otoliths. And so mesopelagic fish, they're not going to experience winter versus summer or food shortage over a large season. And so we don't really know how or why they would lay down increments. Another reason is that age and growth can become decoupled. And so that just basically means that over time, their growth rate is going to change and kind of level off. And if we were to plot their age versus their growth, it's going to give us like a plateau on a curve. And then for otoliths to even be useful in aging at all, those increments need to be regular. So as I said, we try to assume that they're annual. But if they're not laid down in a regular determinable pattern, then there's no point in looking at them because we're not going to be able to determine their age from it. And then the otoliths also have to continue to grow as the fish does. So if the fish keeps growing and the otoliths stop growing, then we're going to have this pretty big disparity on what our estimated age versus length is and the true age versus length. So in order to kind of counteract some of those challenges, we use validating methods on these age assessments. And so validating just makes sure that we understand the time frame that increments are laid down and that they have some sort of annual pattern. And so the first validating method I use is the length frequency analysis. So I'm not going to get super technical with this because it can get kind of boring. But in general, it takes a large amount of length data and plots the frequency versus the time of year. And so this length frequency plot shows a thumbprint emperor, which is a type of fish. And you can see here in the beginning of 2015, we have two different age classes, or what the program assumes, two different age classes. And throughout the year, we can actually see that age class grow in length. And then at the beginning of 2016, we get that very similar pattern of year one age class just around length 10 and age two length class just around length 20. And so this can also be used to kind of backtrack. So if we backtrack age one, we can see just right around here in April is when they started collecting with very short length classes. And so we can use that information and kind of gauge when spawning occurred, potentially around March, as well as tracking changes in size classes or annual mortality, anything that we can kind of track using large amounts of length data, we can use on a length frequency analysis. And then the second method I use is the marginal increment analysis. And so once again, not going to get super technical, but this basically uses length measurements from the otolith. So the marginal increment is this last increment or this last kind of band between the previous dark band and the edge of the otolith. And when we understand that marginal increment and we plot it against the month or even the quarter or time of year that that fish was caught, we can kind of see when all of these annual bands are being laid down, if they're all being laid down at the same time of year. And that would indicate that spawning also occurred all at the same time in 1 year. And so that kind of helps us understand, are these bands laid down annually, biannually, any sort of pattern we can kind of determine from this marginal increment analysis. And so I've rattled on for about 40 minutes with all of my research, but why is any of it important? So I'm going to kind of use this photo as an example. So this is Dr. Kerstetter with an Escalar, and you can see this is a pretty massive fish. Even though it's a big fish, we have no idea how old that fish is or how long it took to get to that size. And so without that information, we can't properly understand the fish stock for this particular species. There's a really high potential for overexploitation, especially if these fish are slow growing and really long lived. So if this is potentially an 8 to 10-year-old fish, we're going to see a time lag in the population rebound. So it's easier to take small fish that grow fast because their populations rebound or kind of grow back within the next year or two. But fish of this size that we don't know how old they are, if they are longer lived, we're going to overexploit all the large individuals at the beginning, and then it's going to take potentially decades for that population to reach the initial size. And so just overall understanding how long it takes a fish to grow, their growth rates, we can track their potential age and length at sexual maturity. And all of that kind of goes towards setting limits or managing certain fisheries, which is pretty important so that we don't overexploit these understudied fish species. So that kind of wraps up what I was going to say with my presentation. The last thing I want to mention is a lot of primary research went into this. And so I'm more than happy to share any of the primary literature that I used. Most of this came from my thesis proposal. So if anyone is interested in any of the sources, any of the scientific literature I used, you're more than welcome to reach out to me and I can share all of that with you guys.
Tiffany, that was awesome. I'm serious. I was actually totally fixated, and I'm not. I always joke around. I'm not. I don't have the science side of the brain over here, and I was totally in your presentation. So I appreciate that. Now I just want to open up to the audience for any questions. There is a Q&A section on there. If you want to put your questions in, I can always allow you to talk to as well. We do have a question from Rhonda Cook. Great job explaining concepts in layman's terms. I'm really enjoying this talk. Thanks, Sydney. When these fish ascend at night, how fast, slow do they go to avoid the bends or pressure issues? And does the oil stink?
So that first question about the ascending, it takes pretty much all night. So over that entire even a little bit longer than a 12-hour period, they're going to travel potentially 1,000 to 1,500 meters. So they're not going to travel at a speed that's going to hurt them purposely. So like when we bring up trawl nets or long line nets, we're trying to bring them up as fast as we can onto the boat so we can process that sample. I don't know exact migration times. I believe it would probably depend on the depth from which they're traveling as well, but that's a great question. And do the oil fish stink? All of the fish stink to a certain extent, but I wouldn't say any more than a normal fish would stink.
That's probably pretty accurate on there. So Rhonda says, thank you. Anybody else have any questions? Oh, we got another question here. Another question. Abby Kerr said, this was brilliant. I worked as a fisheries observer collecting this data, and I really enjoyed hearing the details behind this field of research. Great job. So that's awesome. I have a question for you, Sydney, from my perspective. What did you say when Dr. Kerstetter brought to you this, potential thesis project?
Well, originally we wanted to be able to present something at the undergraduate student symposium my senior year, which ended up being spring of 2020, which didn't happen. So originally, yeah, he was like, I have this project. It was started way back when. Originally, I was focusing more on the data side of it. So what we wanted to present to the undergrad student symposium was more of those length frequency analysis. So taking the data from 1992 to 2020, putting them in those length frequency analyses, and being able to present that length data, showing whether growth is annual or not, or kind of understanding the sizes of the fish populations that we're catching. But it wasn't until I kind of really got into it that we started with the otoliths and the sectioning. And there was actually a graduate student before me, Jess. She did age and growth of yellow stingrays. So she used vertebrae, and she was the one that taught me how to use the saw and all of that stuff. So it's been a learning curve, but it's been a lot of fun.
Oh, I bet. That's cool. Well, thank you for answering that. Now, any other questions from anybody in the audience too as well? Feel free to jump in and ask any questions. Like I said, I can open the mic up to it as well. And you don't mind me sharing your email and information, do you as well, Sydney?
No, that's fine. Perfectly fine.
Now tonight's recording has been, or tonight's lecture is being recorded, so can share it out to everybody here on the YouTube HCAS YouTube channel as well. So, any other questions? Quiet group tonight, but I think you covered it. Just oh wait, this is from Amy Carroll from Rhonda's Computer. For the future, are there ways to create artificial environment to have the fish be closer to the surface and easier to track their change?
That is a great question that's honestly pretty difficult to answer. I'm not an aquarist, but I'm sure in some aquariums they were able to relatively, I wouldn't say recreate entirely the mesopelagic environment, just because of all those conditions. But I know in terms of temperature, that's definitely something you can control. In terms of light, that's also something that we can easily control. It's kind of that pressure aspect of being able to exert potentially 100 atmospheres of pressure onto these fish in an environment that wouldn't normally have that environmental condition. I'm probably not the best equipped to answer that question, but I feel like that would be really interesting, kind of an artificial environment.
Awesome. All right. Well, if there's not any other questions tonight, I know you guys are typing away, but just tell me you have a question. I can open it up. But if not, what are you doing? Wait, it's challenging and important, Rhonda and Amy said. Okay, so all right. Well, I think that's it. So I don't have any more questions coming in, but thank you again for agreeing to do this. I think super important for other students, grad students here at NSU and across, basically across the world, who are interested in marine science to hear, or even our biological science students as well, who are interested in hearing what some of our graduate students are doing. I think you did a phenomenal job in your presentation. I want to thank you and thank the audience for attending tonight. There will be a few more virtual lectures this fall, a couple others in the arts and sciences, and then we'll continue again this coming spring. But I'll share Sydney and my contact information, so if you have questions about the graduate programs, I'll be happy to answer that as well. All right. Thanks, everybody. Have a good night.